everyone, my name is Natalia. I work as a post-bachelor in Dr. Kastner's lab. I've been working there for about a year and a few months now, and next year I'm heading off to medical school.
Hi, my name is Oskar. I also work at the NIH in Dr. Kastner's laboratory. I'm a postdoc, and I did my PhD in Germany at the Max Planck Institute. So we're going to talk to you about new discoveries in diagnostics and DNA sequencing today. And here you see our outline. So first I'm going to give a very broad overview about cells, chromosomes, and DNA. And if you already know everything about DNA, then you can just take a nap, but please wake up after 20 minutes to ask really tricky questions to us. Then Natalia is telling to you about missense mutations and how we can detect those mutations. Then I come back and I talk about a new type of deletions, and it's not new type deletions, but we now can detect this type of deletions, this type of mutations called deletions and duplications. And then Natalia is telling you about the ADA2 protein activity, because she's the expert on this assay. And finally, I give you an outlook, what we think the future of genetic testing will be like. It will be whole genome sequencing, I already spoiled that. Okay. Okay, so this is the human body. I think you can all recognize that. And our body consists of different small building blocks called cells. And in total, we have 200 different types of cells. So they all have different functions. And here I just show you a few of them. For example, fat cells, brain cells, heart muscle cells, or red blood cells. All right, what do you think, how many cells does our human body have? Any guesses? How many? Good point. So in total, there are 37 trillion cells. Actually, they spoiled it already, no? Okay, 37 trillion cells. And in each of these cells, when we zoom in a little bit more, there are 46 chromosomes in each of these cells. And here I chose this picture because it's very beautiful stained. They usually don't have this color, the chromosome. They just stain on this picture. So and usually, we have in each cell, there are 23 pairs of chromosomes. You see the 23 pairs. Pair number 1, for example, you get 1 chromosome from mom and 1 from dad. So then you have 2 chromosomes number 1. The same is true for the pair number 2, 1 from mom, 1 from dad. For the last chromosome, which is labeled here with X, for the last pair of chromosomes labeled with X, when you are female, then you have 2 X chromosomes. When you are male, then you have X and Y chromosome. And these chromosomes, when we zoom in a little bit further, so here you see the cell and the chromosomes within the cell, and these chromosomes consist of a very long strand of DNA strand. And this DNA strand consists of only 4 letters, the A, C, G, T, C, A, A, and so on. And on this DNA strand, there are these very important structures called genes. And when we write down all the letters of one cell, of all the chromosomes in one cell, there are 3.2 billion letters. So 3.2 billion times A, C, G, T, 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 and so on. Okay, and here, very, very useful information for you. When you would write down the 3.2 billion DNA letters, you could fill 200 New York City telephone books. You could also write 800 Bibles. And if some person would be willing to do that, you could, when a person would type 60 words per minute, 8 hours a day, that would take 50 years to write our whole genome, 3.2 billion letters. And even more important information, when we would connect all the DNA strands of all our 37 trillion cells, then we would get a strand that is as long as we would go 6,000 times to the moon and back. So very long and complicated. And with that, I hand over to Natalia.
Thank you, Oskar. So, much like Oskar said, a gene constitutes a specific part of the DNA. Thank you. There are specific parts to a gene, so I'm going to try to point it at it on this side. A gene is made up of exons and introns. Exons are parts of the gene that are ultimately expressed, and introns are spacer regions between exons. And I'm going to get into a little bit more detail later on. A DNA gets transcribed into mRNA, and then that mRNA gets translated. So when you have your mRNA, each 3 letters of the RNA gets assigned an amino acid. And amino acids are basically what makes up a protein. Proteins have many different functions, but they're basically the building blocks of life. And as you can see here in this example, each exon corresponds to a specific domain in the protein. So I want to take a moment just to have an analogy, just to make this easier to understand. So let's imagine that you're like sitting down on your computer and you're writing the first draft of an essay. So you're sitting down and you're typing all of these letters. Now remember that in the world of genetics, you can only type A, T, G, and C. So that first draft of your essay is your DNA, just a bunch of letters, in a specific order, of course. Then when you then go back to that essay and you're getting rid of unnecessary letters that don't really add much to your story, then you're undergoing this RNA processing side of this process. And then you then take 3 letters from that mRNA strand from your draft, and you assign a word to it. That word is an amino acid. And when you have a beautiful long stretch of words, you make a very structured sentence. And that, in this case, is a protein. And much like each sentence has a specific meaning, proteins have a specific function. And so let me just now talk about missense mutations. So when you're first typing that draft, let's say you type a G instead of a T, right? That base pair change in your DNA is what is called a missense mutation. And that's denoted by that little arrow right there. And so, as you can imagine, if you end up misspelling something in your DNA, that can alter a word, and that can alter an amino acid in your protein. And that is going to be directly, that can directly affect your protein because you have now a different amino acid. And so that can either make that protein work better or worse. So now let's talk. There's actually quite a few missense mutations in DATA2, but now I want to make this brief overview of what DATA2 is. So I'm sure you all have heard that DATA2 is considered a monogenic disorder. So does anyone know what monogenic means? Great, so awesome. Monogenic means that there is one specific gene that is responsible for a disease or deficiency. So you're nodding, you should have answered that question.
I'm a biology.
Oh, cool.
I don't know this.
So then you can correct me if I say anything wrong here. Then. Yeah, so exactly. So monogenic is when you have one specific gene responsible for a disorder. So in the case of DATA2, that gene, in the case of deficiency of the adenosine deaminase 2, even I get it mistaken sometimes, the gene is adenosine deaminase 2. And DATA2 is considered an autosomal recessive disorder. So like Mandy mentioned a little bit before, in the autosomal disorder, recessive disorder, you need to have 2 copies of your gene mutated for you to be considered an affected individual. So in this case, the affected individual is here, and I used a mutation, and I will get into what these letters and numbers here mean later on as an example. So this affected child has 2 mutations, R169Q. And the parents of this child are carriers. That means they have 1 functional copy of that gene, and they have 1 mutated copy of that gene, as you can see here. And so, oh, question?
I have a question about that. You know, like you have it in the picture right there, the marks where it's the mutation. If that marks, like when one of them goes in the bottom part and then the other one goes in the upper part, the location of the mutation, it makes different?
It's the same location. So you're giving.
Exactly the same location?
Yeah.
No, you can also have in different mutations, but same in the ADA2 gene, different mutations. But if the child gets one mutation from dad and the other from mom, even if they're different, they are affected.
Yeah.
But it has to be in the ADA2 gene.
Yeah, in the same gene, but in different location of the gene,
You
and
would,
don't need to be exactly in the same
no,
location.
no.
Yeah, but the mutation that you have as a carrier, as a person, you're giving that same mutation to your child. Sorry, I thought that's what you meant. The mutation doesn't change by if you have a child.
I always thinking that maybe I hear something that I have it in one location and my husband has it in another that maybe is giving him a less symptoms than everyone else?
Right now, we can't really, we can't really, like Mandy said, the spectrum of data too is so wide, we are not right now able to specify what mutations give a sort of phenotype. So if you have a specific mutation, your husband has another one, that combination, we can't really say whether it's going to make the disease better or worse. And if you have any...
That's correct. So it's very hard to tell what specific mutation would cause what symptoms or what manifestations. That's very hard to tell right now.
It's like if you can do a comparison with all the people who have the data to exactly in what locations of the gene they have the mutation and see if that gives to someone a lot more of symptoms and to another one less of.
That's a very good point. But we had this example here with this family. They have the same mutation, and all three siblings have different manifestations, even it's the same mutation. So it's other factors contribute, like environment or other mutations somewhere else in the gene.
Yeah, you just see, okay, what is the similarity? What is the position, the location of that?
Yeah. Yeah. Good question.
Yeah.
Good question.
Go ahead.
Are we allowed to ask questions now? I don't want to interrupt your presentation, but
This is very informal. Go ahead.
this says in the marriage among relatives, he the cause of EBA2. But he's married to his cousin, fraternal cousin. And so could that be the cause for the EBA2? Because he's a carrier, his wife is a carrier, and they have, a kid that's, affected by the disease. The one that passed away when his son was 14 years old. So could, like, marriage among relatives be the cause?
So marriage among relatives is not the cause, but when you do marry someone who is part of your family, you do have an increased risk of having mutations in the same gene. So that's called consanguinity in the world of genetics, and so that's basically it. You just happen to be that the person who you married, because you're part of the same family, you have a higher chance of having mutations in that same gene. That's basically it. So it's not the cause, but it does increase your chances of having an affected child. Yeah. So, like I was saying, any more questions? Oh, sorry. It's okay.
You keep saying the mutation could be for the better or for the worse. Yes. So we hear about all the worst things. What is the better thing then?
So not in this case, unfortunately, but there are some cases where if a mutation, so it depends on the function of the protein. Let's say a protein is meant to make you break down a specific food better. It breaks down a specific food, like fries, right? You eat fries and then you break it down to specific parts. If you have a specific mutation in that protein that enhances its activity, it's then able to break down fries in a faster way or in a faster way that gives you more nutrients. So that would be an example. Right now, I guess because I'm in the spotlight, I can't really think of specific disease.
That's one other famous example is sickle cell anemia.
Yeah, okay.
So when you have sickle cell anemia, it's a very common disorder in African regions. And when you have only one of the chromosomes, for example, the one from your mom, when you have a mutation on that, then you are basically intolerant against malaria. So you cannot get malaria. So this mutation would help you to avoid malaria. But if you have two, then it's a disorder and you have problems to transport O2 to your different body parts. So this is another very famous example.
Okay. Better than my fries example. So, any more questions? Okay. So, like I was saying, oh, sorry, go ahead.
What's funny is my parents are both careers, and I'm one of 5 siblings. I'm the only one who has mutations as well. I have it. My husband's a carrier, and all of my children have it as well. Is that the reason is that the more dominant one, this deficiency?
So the deficiency is still considered a recessive disorder. However, because you're affected, so you have two mutations, a mutation in both copies of your gene, and now your husband has a mutation in one copy of your gene, then your child is at an increased risk of getting that disease. So, if let's just go back to that example, and I can expand on that a little bit later on. So here, carriers have one mutated copy of the ADA2 gene. So you have a 1 out of 4 chance of having that disease. So because you have two copies, you give one to each. You give one to each child, 1 out of 4, so you have 4 possibilities here. 1 out of 4 is that you're affected, and then alternatively, you have a 1 out of 4 chance that you're completely unaffected, which in the science world we call it that person wild type. And so you have a 1 out of 2 chance because you have 4 possibilities. You have 2 out of those 4 possibilities. You have 2 possibilities that you're going to give at least your child is going to receive at least 1 affected gene. So that means you have a 1 out of 2 chance that your child is going to be affected. So now that changes. So you're this child. You have 2 affected copies of the gene, and your husband is a carrier. That means, Oskar, can you do the math real quick? That it would be 50 chance?
50%.
50% chance that your son will be a carrier, yeah. Okay, so this, in case you've ever gone to at night in your room, you're ever wondering, gosh, what does 882 look like? What does the coding region of 882 look like? We put together the slide full of letters just to show you what the coding sequence of this gene is. But in all seriousness, it just also goes on to show you that there are quite a few base pairs or letters in this gene that have the chance of being mutated. So in fact, now that this is again 882. At the top you see 882, the gene 882, and at the bottom it's the protein. Like I said, these boxes are the exons that come together to form the protein. Right now we know of 70 mutations in 882. Now when we first had the Data 2 conference, we did not know that much. So we're much like in the clinical side, we're still learning more phenotypes and of how patients respond to medicine. In the genetic side, we're still discovering new mutations as well. And the majority of these mutations are missense mutations, like I mentioned, but other mutations also exist, like frame shift deletions, duplications, and it's something that we're going to get into a little bit more later on. And additionally, I wanted to highlight the fact that, yes, these mutations do affect the protein, but there are some mutations that are present in your DNA that are not present in the protein, but still alter protein function. So now to the more interesting side, or at least for me, this is what I mostly do in the laboratory. I do all of the diagnostic sequencing of patients that our clinic sees. So in a nutshell, what my job involves is I do an experiment where I add a specific color to each of your nucleotides. So you have A, T, G, C of your nucleotides in your DNA. So an A gets green color, a C gets a blue color, a G gets a black color, a T gets a red color. And then after I do this reaction here at the bottom, I put it in this handsome machine here that we have. We just got it last year. We got it upgraded. I'm very excited about it. Yeah. And that machine has a laser that detects each color, and it outputs a graph that looks like this. So this looks like a chromatogram. And when we're doing Sanger sequencing for specific genes like 882, I see all of these peaks that correspond to a specific base pair in your DNA. Now here, I highlighted this square right here. Like I mentioned, you have 3 letters in your mRNA that code for a specific amino acid. So here, these 3 letters, CGG, code for this R, which stands for arginine. So this C right here in the middle is at position 506 in your gene. And this CGG then codes for arginine, which is at position 169 of your protein. Okay? So a person who does not have any mutations in 882 then looks like that. That person, again, is called a wild-type individual. And they have a CGG, for this specific example, a CGG from mom and CGG from dad. And so they have like these beautiful peaks here that correspond to that specific base pair. Now, a person who is a carrier has then, for this specific mutation, they have a misspelling in this G that turns into an A. So this misspelling changes the arginine, the amino acid at position 169, to a glutamine. A carrier then has inherited this CAG from the mom or the dad, or in the CGG from the other pair, the wild type reference. So when I analyze these sequences on my computer, I see 2 peaks for a person who is heterozygous for this mutation, which means that they have a mutated copy and a wild type copy. Okay, so 1 peak corresponds to the A, which is the mutated letter, and then the 1 peak corresponds to the G, the wild type letter. And now a person who is, in this person who is heterozygous, they would then be considered a carrier if they don't have another mutation. And now a person who inherited the same mutation from both parents would then be considered homozygous for that mutation. So again, this person has this altered base change here, the A and the A, and what I see on my chromatogram is one peak that corresponds to this mutated A. This person would then be considered homozygous for this disease, and this would be an individual affected with DATA2. Hand it off to Oskar now.
Okay, besides this one-letter spelling mistakes, there are also other mutations, and they are called deletions and duplications, and I'm going to talk to you about those now. So here you see a chromosome, and you can see that there are two breaks in the chromosome on the very left-hand side, which causes this piece in the middle, the slightly red one, to be gone afterward, and the chromosome gets shorter. And this can also happen in the ADA2 gene. So this is again the sequence of the ADA2 gene, all the letters. And sometimes we see a mutation that looks like that. So a piece is missing, basically. This is a deletion. There can also be duplications, and this is basically the red and green parts get duplicated because of two breaks in the chromosome, and then this piece gets integrated again, and you have a longer chromosome. In the case of ADA2, the black part here is duplicated and just added in the red area again. So this happens also. And 2 years ago, we were not able to identify those types of, most of those we couldn't identify with the method that Natalia just explained. But we have now two new assays that can detect those mutations as well. And the first one is called multiplex ligation-dependent probe amplification. I call it MLPA because it's easier. And no worry, the most complicated part of this assay is its name. But the assay itself is very easy and is very successful. So here you see a healthy individual of whom we know that there are no deletions or duplications in the ADA2 gene. On the right-hand side, the slightly orange part, this is just locations on different chromosomes of which we know that they are in two copies. So and you see that this line, basically the green area, the orange area with the green bars, this is two copies. And in this individual, all the ADA2 gene, the exon 1 to 10, has also two copies. So this is a healthy individual, no mutations. But here we have one patient of whom we know that he has a deletion of exon 1 and intron 1. And you can see that all the control areas and other chromosomes are at two copies. All the exons from 2 to 10 are at two copies, but intron 1 and exon 1 the signal is lower. That means there's a deletion, only one copy, because the other chromosome of ADA2 is still fine. The other gene is still fine. Only one lacks exon 1 and intron 1. So this was our positive control. And before we had this assay, we had several families where we couldn't find the second mutation. So we used this assay to look for mutations that are not spelling mistakes, but deletion and duplications. And here I show you a family with 3 daughters. And we knew that the daughters, all 3, had a pathogenic, a disease-causing mutation on one chromosome, but we couldn't find the second mutation on the other chromosome. But we knew that they had DATA2 because they clinically had DATA2. So we did this assay, and you can see that daughter 1 and daughter 2, the results here, have a deletion of exon 7. And this explains why they have DATA2, because the other chromosome now is also mutated. I don't show you daughter 3 because the results were really ugly, so we have to repeat that. And here I show you another family. You see that dad is a carrier, father is a carrier. He has one wild-type chromosome and one mutated chromosome with a T129P mutation. And mother, we know that she was wild-type on one chromosome, but we could never find, so we know that daughter 1 is affected because she has DATA2 symptoms. And we know that she inherited the T129 mutation chromosome from her dad, but we couldn't find the second mutation. When we did this assay on daughter 1 and on the mother, we saw that exon 8, 9, and 10 were deleted in both of them. So that means mom is a carrier, so we can substitute the question mark with exon 8, 9, 10 deletion for mother, but she's only a carrier, though she's not affected. And daughter 1 has now 2 mutations, the T129P and the exon 8, 9, 10 deletion. So she's affected. And since a few weeks now, we use another assay, which is also a complicated name, digital droplet polymerase chain reaction, in short, DT-PCR. And this is an even more sensitive method, but it's also used to detect duplications and deletion mutations. And here I show you our latest result. You can see that the first 3 dots or squares, the black ones, are no DNA. So we didn't put any DNA in the tube. That means we don't get any copies, so 0 copy numbers for ADA2. Makes sense, because no DNA, no copy numbers. The next 3, the gray bars and the gray squares, are healthy control. So we get 2 copies of exon 8, 9, and 10. We only tested these 3 exons, we didn't test the other 7 exons. But now we use the patient from the slide before, I think it was a mother, who has on 1 chromosome a deletion of exon 8, 9, and 10, and you can see that the copy number goes down to 1 copy only. So we can now use this assay, which is more sensitive than the MLPA assay, and this is what we are going to do in the future. And with that, I hand over to Natalia.
Thank you. So Oskar and I both mentioned mutations and ways to detect these mutations in the DNA. So another way that we can also detect for adenosine deaminase deficiency, adenosine deaminase 2 deficiency, is by testing the protein activity. So we use this assay. It does not have a fancy name like MLPA. We just call it the ADA2 protein activity assay to measure ADA2 activity. So out of the many blood tubes that you give to us in the clinic, is one tube called the serum separating tube. So you first get the tube drawn, and then I take it to the lab, I spin it down, and so that way I can collect plasma that's present in your blood. Now, these are the red blood cells, and this is the plasma. This is what the blood is made up of. I throw away the red blood cells. I'm not interested in those. I want to get the plasma, which has the ADA2 protein and other proteins in it. And I use that plasma to test for protein activity in this assay. So it's basically a color-based assay. The purple color is indicative of increased protein function. So the brighter color you have, the more ADA2 protein is active or that works. So here, as you can see, we also have a water control in our assay. So in this water control, we had no plasma from no individuals. We just have water, just so you can see that there is some background fluorescence here. But now let's compare this water control to DATA2 patients. It's very similar, right? So there is almost no ADA2 activity in DATA2 patients. That is because you have 2 mutated copies of your gene, copies of your gene, so you don't have protein function. Now carriers, they fall sort of in the middle ground between healthy controls and DATA2 patients. That's because you have 1 functional copy, so you are still able to convert that purple color, convert that blank color to a slightly purple color, but not so much so as healthy controls. And now, as you can see, these other individuals that we're testing, there is some variability in the purple light or the purple color that we see here. That's because every person is different. In some of you, ADA2 works a little better than others, and also that's also correlated to age. But it's important to know that DATA2 patients do have a remarkable difference compared to other individuals who don't have mutations in both copies of the gene. Yeah.
Is there a connection between the DATA2 level and the 2 levels in the blood and risk of stroke? Because I once read an article that it was not much patients, and they said when that's about 1, I think the risk of stroke is low, but the each one there was bigger risk. But I don't know for the full population.
I know this paper or this article. They claim that the lower the ADA2 activity, the higher is the risk for stroke. So we don't see that in our clinic. So I think it's hard to tell. Even for carriers, even if you have 50% of activity left, we don't know exactly if this would predispose to any symptoms. So this is still under investigation. But I know this article, and that's true. Yeah.
So, just to go off of that, just to diagnose or help diagnose someone who has DATA2. So, for example, before we had this MLPA assay, we only had Sanger sequencing. We also used this assay to help make that diagnosis. So we can't really correlate it to stroke yet, but this assay is very useful just to tell us that yes, this person has that attitude. They have no level of protein activity. Go ahead.
Okay, so mom and dad gave it to the child. Where did mom and dad get it? From their mom and dad, generations back? I mean, is this random? How does this work?
You can take it.
Very good question, very good question. So we see that the frequency of these mutations in the population are not very low. So many people walk around with one mutation. They are fine because it doesn't cause disease in one mutation. But they potentially, if they find a partner who has another mutation or the same one, then the child would be affected. So it's just by chance. Also, there are also mutations that happen, we call it de novo. This happens when in sperm cells or in the egg cell, sometimes there's a mistake in the writing of this DNA, and then you can get a new mutation, which by very unlucky cases can be also a pathogenic one, and this happens also. So it's these both factors.
Yep.
Okay. Any more questions? Then I want to end with a very brief outlook of what we think the future of genetic testing will be, not only for DADA2, but for basically any genetic disorder. It will be whole genome sequencing. So in 2001, the first human genome was published. It's already 17 years ago. It's a very long time. And it took 15 years to get this, to read these 3.2 billion letters of the human genome. And it was very expensive. It cost $1 billion. So, but over time, with technologies improved and getting better and faster, we are now at the state of getting a sequence, a whole genome sequence, 3.2 billion letters for less than $1,000. And it doesn't take 15 years anymore. It takes like less than 1 week. So you can get your DNA drawn, or your blood drawn, get DNA, and get your whole genome sequence. And the advantages of whole genome sequencing are many. So I just list a few here. We can find this deletions duplication that I just told you about that are tricky to find when we just do the Sanger sequencing. We can find those easily when we do whole genome sequencing. We can also find mutations that are not necessarily in the important parts of the gene, like the exons, but also in the introns or somewhere completely else. And that might affect also the ADA2 gene. We can find so-called somatic mutations. Somatic mutations is another thing that is very tricky to identify, because this means that you have a mutation only in a few cells of your body, in not all of the cells, but only a few cells. And if this is your sperm cells or your egg cell, then this can be dangerous, and you can be tested on your blood. We don't find a mutation, but you might have the mutation in your egg cells. This would be very unfortunate. This happens, unfortunately. And it will be really fast. Within 1 week, we can get the result of whole genome sequencing. The problem is that we only know about basically 2% of the whole genome yet what they do. We don't really understand the other 98% of the whole genome. So this is still research ongoing, but as already Albert Einstein said, if he would knew what we are doing, it wouldn't be called research. So we are doing our best, but it takes some time. Thank you very much.
Thank you.
Absolutely.
Still that research cost. I'm serious. Is it in the millions of dollars? Because I'm fundraising, so I need to know what kind of money is needed for this research.
I mean, I can speak, it's definitely in the millions, but we are a small group. We might not need millions, but there are many groups working on data too. So
All over the world.
exactly. So it's very expensive.
Lots of money.
Yeah.
Exactly. So, yeah.
Go ahead.
And the siblings of this patient, when you do fertility carrier or Bethesda, you don't go Bethesda baby on that. You didn't want the whole genome sequencing for the test, or it's just tied into that. My point is, the other kids can have another misspelling with another sickness, or just because they are just carriers there, don't have the other ones. So you don't see the other ones, right? In the carriers, you don't test for the other ones, just basically for the other ones?
When we're testing siblings of affected individuals, we sequence the 8A2 gene. So we look through the whole gene to see to look for mutations. It's always the case that if they do have one, they would have the one that is present in either parent. So we've never had a carrier or sibling who has another random mutation 88.2 that's not present in the family. But we do look for the specific gene. We don't do whole genome sequencing or whole exome sequencing. No, we just look for Cs here at 88.2.
Okay, I suppose if the family goes like a Ehlers-Danlos syndrome, that one, it wasn't tested, right?
Correct.
It's just specifically on the
Specifically, exactly.
Okay, we only have time for a couple more questions.
You know, if you've been diagnosed with DARTA2, because all four of my children have, but one of them's more severely affected than the other three, is that because there might have been a slight difference in the mutation they received, say from my husband or from myself?
So you say you also have DARTA too. That means you have 2 mutations. Do you know if these are the same mutations or if they are different mutations? Because if you would have the same mutation, let's assume that, then your children have all the same mutation from you and the same mutation from your dad, from your husband, from their dad. That means they have always, they all 4 have the same mutations. Is it correct? Your husband is a carrier, so he only can give one mutation to all 4. You can give, when you also have only 1 mutation, or you have 2 mutations but the same one, then you can also give only this type of mutation. So your children should have all the same mutations. If they are now different in severity or in manifestations, then this is something we cannot explain yet. And this is what we see. Even with the same mutation, different findings in the patient. So when we try to understand what this could be, what the reason could be for that, but that's when we do whole genome sequencing. We might find other mutations somewhere else in the genome who contribute to that. But this is very tricky to find the association between mutations somewhere else in the genome that also make DARTA2 more severe or less severe. This is ongoing as well. But so far, we cannot tell you about other mutations that have an effect on the DARTA2 severity.
Okay, last question.
Blood work is not done. Blood work is not done at the NIH, but is done through an outside source and then is sent out. Do you get all of the findings, and is the control the same of the testing?
So for Sanger sequencing, we do get mail-ins, so people from all over the world, if they want to get tested for DARTA2, we get their blood. And yes, we test it in the lab. So for Sanger sequencing, you don't need a control, let's say, because when you just extract the DNA from your blood sample and you run it through that experiment that I did, you add the colors, you run it through a machine, and then the reference that I align that sequence to is known to everyone. So that's the healthy control reference that individuals in other facilities all over the world have access to. So in that case, there's no variability in between controls for Sanger sequencing specifically. If you're doing MLPA or other types of duplication assays, other institutions can do that as well. They have their own controls, but you know it's a healthy control, so we can basically they are still able to diagnose individuals like that. Did I answer your question? Yeah.
Okay, thank you both so much.
Thank you.
It was great.